So here's something worth thinking about as you settle in tonight. Right now, as you're lying there, maybe adjusting your pillow, maybe pulling the blanket up a little higher, there's this thing sitting inside your skull that is, without exaggeration, the most complicated object we've ever found in the known universe. And I don't mean complicated the way a smartphone is complicated or the way a jet engine is complicated. I mean complicated in a way that has humbled every single person who has ever tried to fully understand it. Your brain. About 3 pounds of wet, pinkish-gray tissue, roughly the texture of firm tofu, and it is running everything. Every thought you're having right now, every sensation of the sheets against your skin, that little background hum of whatever you were worrying about earlier today, the fact that you can understand these words at all, that's all happening in there. And the wild part, the thing that should honestly keep you up at night, except that the whole point of this is to help you drift off, is that for most of human history, we had almost no idea what it did. Let's start there. Let's start with how spectacularly wrong we were for how long, because honestly, that's one of the most fascinating parts of this whole story. The ancient Egyptians, brilliant as they were, these are people who built the pyramids, who developed sophisticated mathematics, who performed surgeries. They thought the brain was basically packing material. Stuffing. When they mummified their dead, they had this whole elaborate process for preserving the body for the afterlife. The heart got carefully removed and placed in a special jar, because the heart was everything to them. The heart was where you thought, where you felt, where your soul lived. But the brain? They stuck a hook up through the nose, scrambled it around, and drained it out. Just threw it away. The most complex structure in the universe. And they literally scooped it out and discarded it like it was nothing. There's something almost poetic about that level of misunderstanding, and they weren't alone. This idea that the heart was the seat of the mind persisted for a remarkably long time. Aristotle, one of the greatest thinkers in all of Western history, writing around 335 BC or so, was firmly in the heart camp. He looked at the brain and thought, well, it's kind of cold and bloodless compared to the heart, so it must be a cooling system, like a radiator for the blood. The heart was warm and active and rhythmic, and it seemed to respond to emotions. Your heart races when you're scared. It pounds when you're in love. So, of course, that's where thinking happened. The brain was just there to keep things from overheating, and you can see the logic, right? If you didn't know anything about neurons or electrical signals, if you were just observing the body with your own senses, the heart really does seem like the more important organ. It's loud, it's central, it responds to everything you feel. The brain just sits up there, quiet, doing nothing obvious. But not everyone agreed with Aristotle. And this is where the story starts to get interesting. There was a physician named Alcmaeon, working in the Greek colony of Croton in southern Italy, probably around 500 BC, so actually well before Aristotle. Alcmaeon did something that was pretty unusual for his time. He dissected things. He dissected animals, and possibly human bodies too, though the historical record is a little fuzzy on that. And he noticed something. He noticed that there were channels, what we'd now call nerves, running from the eyes and the ears directly into the brain, not into the heart, into the brain. And he made this remarkable leap. He said, essentially, the brain is the organ of sensation and thought. He was right. 2,500 years ago, this guy figured out the basic truth. And then Aristotle came along a century and a half later and said, nah, it's a radiator. That's how science works sometimes. It's not a straight line. Someone gets it right, and then the next brilliant person gets it wrong. And the wrong answer wins for centuries because the person who said it had a bigger reputation. Now, the person who really started to turn things around, at least in the ancient world, was a physician named Galen. Claudius Galenus, born around 129 AD in Pergamon, which is in modern-day Turkey. Galen is one of those figures in the history of medicine who is so influential that his ideas dominated European thinking for well over 1,000 years. And Galen was a fascinating character. He started his medical career as the physician to gladiators. Think about that for a second. Gladiators. He was patching up men who had been slashed and stabbed and bludgeoned in the arena, and that gave him an extraordinary education in human anatomy, because he was seeing the insides of living people in a way that most physicians never did. He saw what happened when a nerve was severed. He saw what happened when different parts of the body were damaged. And he became absolutely convinced that the brain, not the heart, was the center of the nervous system. Galen did experiments. He would press on the exposed brains of living animals. And yes, this is uncomfortable to hear about, but it's part of the history. And he observed that pressing on the brain could stop movement, could alter behavior. He traced nerves from the spinal cord up into the brain. He identified that there were different nerves for sensation and for movement, which was a genuinely brilliant observation. But here's where Galen went sideways. And this matters because his mistake shaped how people thought about the brain for the next 1,500 years. Galen was fascinated by the ventricles. The ventricles are these hollow chambers inside the brain, filled with fluid, what we now call cerebrospinal fluid. And Galen decided that the ventricles were where the real action was. He thought that the brain worked by producing and moving around different kinds of spirits, or pneuma, through these chambers. The solid tissue of the brain, the actual stuff we now know does all the computing, he thought was relatively unimportant. It was just the housing. The ventricles were the engine. This is called the ventricular doctrine, and it stuck around forever. Medieval scholars inherited it, elaborated on it, assigned different mental functions to different ventricles. The front ventricle was for perception and imagination, the middle one for reason and judgment, the back one for memory. It was a neat, tidy system, and it was almost entirely wrong. But it felt right. It had the weight of Galen's authority behind it. And for century after century, if you wanted to understand the brain, you studied the ventricles. Here's the thing about the ventricular doctrine that's worth sitting with for a moment. It wasn't just wrong in a trivial way. It was wrong in a way that actively prevented progress because it directed everyone's attention to the wrong part of the brain. The ventricles are real, and they do serve a purpose. The cerebrospinal fluid cushions the brain, removes waste, does some important housekeeping. But they are not where thinking happens. Thinking happens in the tissue itself, in the neurons, in the connections between them. And as long as everyone was staring at the fluid-filled chambers and ignoring the grayish tissue around them, they were never going to figure out how the brain actually works. It took until the Renaissance for things to really start shifting, and even then, it was slow. Andreas Vesalius, the great Flemish anatomist working in Padua in the 1540s, started doing something radical. He actually dissected human bodies himself, carefully and systematically, instead of relying on Galen's descriptions. And Vesalius noticed that the ventricles in human brains looked pretty much the same as the ventricles in animal brains. Sheep, cows, dogs, they all had ventricles. So if the ventricles were where human reason and judgment lived, why didn't sheep have reason and judgment? It was a simple observation, but it was devastating to the ventricular doctrine. Vesalius didn't have a better theory to replace it with, not really, but he had the courage to say that the old theory didn't hold up. And this is a pattern you'll notice as we go through this story. Again and again, someone has the insight to say, this is wrong, long before anyone can say, here's what's right. Progress in understanding the brain has almost always come in two stages. First, tearing down the old idea, and then, sometimes decades or centuries later, building up the new one. So, by the late 1600s and into the 1700s, the ventricular doctrine was fading, and people were starting to pay more attention to the brain tissue itself. But now, they had a new problem. When you look at a brain, even with the best tools available in the 17th century, it's incredibly hard to see what's going on. It's dense, it's wet, it's all kind of the same color. You can see that there are different regions. There's the wrinkly outer layer, the cortex, and there's white matter underneath, and there are distinct structures deeper inside. But what do? How do you figure out which part does what? This is where a really important idea starts to take shape, and it's one we'll keep coming back to because it's central to how we understand the brain today. The idea is localization of function, the notion that different parts of the brain are responsible for different things, that there isn't just one homogeneous blob doing everything, but rather a collection of specialized regions, each handling its own job. And the story of how we figured that out is one of the strangest, most contentious, and most fascinating chapters in the history of science. It starts, oddly enough, with a con man. Well, that's not entirely fair. Franz Joseph Gall was a legitimate anatomist, born in 1758 in what's now Germany. He was genuinely skilled at dissecting brains, and he made some real contributions. He was one of the first to clearly distinguish between gray matter and white matter, and to suggest that the gray matter of the cortex was where the important stuff happened. That was a big deal. But Gall is remembered today mostly for phrenology, and phrenology is one of those ideas that was so spectacularly, flamboyantly wrong that it's become a punchline. But here's what's interesting. The core intuition behind it wasn't entirely crazy. Gall's idea was this: different mental faculties, things like language, mathematical ability, aggression, love, caution, are located in different parts of the brain. So far, so good, right? That's basically what modern neuroscience tells us in broad strokes. But then Gall made a leap that had no evidence behind it. He said that if a particular faculty was well developed in a person, the corresponding part of the brain would be larger, and that this enlargement would push out on the skull, creating a bump you could feel. So by running your fingers over someone's skull, you could read their personality, their talents, their character. Bump here means you're good with numbers. Bump there means you're prone to violence. Bump over here means you're especially loving. It was nonsense. Complete, utter nonsense. The shape of your skull has almost nothing to do with the shape of the underlying brain tissue, and even if it did, mental faculties don't work that way. But phrenology became wildly popular. In the early 1800s, it swept through Europe and America like a craze. People went to phrenologists the way they might go to a therapist today. Employers used it to screen job applicants. It was used to justify all kinds of ugly racial theories. Queen Victoria had her children's heads read. It was everywhere. And the backlash against phrenology, when it came, was so strong that it almost killed the legitimate idea underneath it. Scientists became deeply suspicious of any claim that specific functions lived in specific brain regions. A French physiologist named Pierre Flourens did experiments in the 1820s where he systematically destroyed different parts of animal brains and observed what happened. And his conclusion was that the brain worked as a unified whole. Damage any part of it, and everything got a little worse. But no specific function was completely lost. This was wrong, as it turned out. But it was wrong in a way that seemed like a reasonable correction to phrenology's excesses. So now you've got this tug of war. On one side, localization. Different parts do different things. On the other side, what's sometimes called equipotentiality. The brain works as a whole. And this debate is going to rage for decades, and it's going to take some truly remarkable discoveries to settle it. One of those discoveries involves a man named Paul Broca, a quiet French surgeon working in Paris in the 1860s, and a patient who could understand everything said to him but could produce only a single syllable. That patient's name was Louis Victor Leborgne, but everyone at the hospital called him by the only sound he could make, Tan. And when Tan died and Broca examined his brain, what he found in one specific region of the left frontal lobe was going to change everything. Because what Broca found, when he carefully examined Tan's brain after the autopsy in April of 1861, was a lesion, a region of clear, unmistakable damage, sitting right there in the posterior part of the left frontal lobe. Not scattered damage, not general deterioration across the whole brain. A specific area, roughly the size of a chicken egg, that had been destroyed, probably by a slow progressing infection, or possibly a stroke years earlier. And the thing that made this so extraordinary was the precision of the loss. Tan could understand language perfectly well. He could follow instructions. He knew what people were saying to him. His intelligence, as far as anyone could tell, was intact. He just couldn't speak. He could produce that one syllable. Tan, and sometimes a curse word, which is actually a fascinating detail we'll come back to later, because it tells us something really interesting about how the brain organizes different kinds of language. But meaningful, voluntary speech? Gone. Completely gone. And it was gone because of damage to one specific spot. Now Broca was a careful man. He was a surgeon, an anatomist, someone who believed in evidence. He didn't rush to publish some grand theory based on a single case. But he presented Tan's brain to the Société d'Anthropologie de Paris, and he made a measured claim that this region of the left frontal lobe appeared to be critical for the production of articulate speech. And then, over the next few years, he gathered more cases, patient after patient, who had lost the ability to speak while retaining comprehension, and in case after case, when the brain was examined after death, there was damage in that same general area. By 1865, Broca had accumulated enough evidence to make an even bolder claim, not just that speech production was localized, but that it was localized specifically in the left hemisphere. This was huge. The idea that the two halves of the brain might not be perfect mirrors of each other, that one side might be dominant for certain functions. This was genuinely revolutionary, and the region he identified. It still bears his name today, Broca's area. If you put your finger on your left temple and move it slightly forward and up, you're pretty close to it. Here's what's really beautiful about this discovery, though. It didn't just settle the localization debate, or at least, it didn't settle it cleanly, because science never works that neatly. What it did was crack the door open. It showed that yes, Gall's core intuition had been right all along, even though phrenology itself was nonsense. Different parts of the brain really do handle different jobs, and once that door was open, other people started walking through it. About 13 years after Broca's discovery, a German neurologist named Karl Wernicke, and he was young, only about 26 years old when he published his key work in 1874, found a complementary piece of the puzzle. Wernicke studied patients who had the opposite problem from Ton. These patients could speak fluently. Words poured out of them. But the words didn't make sense. They'd string together grammatically correct sentences that were essentially meaningless, word salad, as it's sometimes called. And these patients couldn't understand speech either. You could talk to them, and they'd look at you blankly, or they'd respond with something completely unrelated. When Wernicke examined their brains, the damage wasn't in Broca's area at the front. It was further back, in the upper part of the left temporal lobe, near where the brain processes auditory information. And that makes intuitive sense, right? If you think about it, comprehending speech is fundamentally about processing sounds and connecting them to meaning. So it makes sense that the brain region handling comprehension would be located near the auditory processing areas. Wernicke's area, as it came to be known, gave us the other half of the language picture. Broca's area for producing speech, Wernicke's area for understanding it. And Wernicke went further. He proposed that these two regions were connected by a bundle of nerve fibers, and that if you damaged that connection without damaging either region itself, you'd get yet another kind of language problem. The patient would be able to speak and able to understand, but they wouldn't be able to repeat things back to you. They'd hear a sentence, understand it perfectly, try to repeat it, and produce something garbled. This condition is called conduction aphasia, and it does exist, and it was a remarkably prescient prediction for the 1870s. Wernicke was essentially building a circuit diagram of language in the brain, decades before anyone had the tools to actually trace those circuits. So let's pause here for a second, because I want you to appreciate what's happening in this period. In the span of about 15 years, from the early 1860s to the mid-1870s, the understanding of the brain has shifted dramatically. We've gone from the brain works as a unified whole to, actually, specific regions handle specific functions, and those regions are connected to each other in specific ways. And if you damage the regions or the connections, you get specific predictable deficits. That's an enormous conceptual leap. And it was driven almost entirely by careful observation of what happened when things went wrong. These early neuroscientists didn't have brain scanners. They didn't have electrodes they could safely implant. They had patients with brain injuries, and they had autopsies. That was it. And from that, they built a surprisingly accurate map of how language works in the brain. But here's the thing, and this is important. The localization story was never as clean as it sounds when you tell it quickly. The brain is not a collection of neat little boxes, each doing one thing. It's more like, think of it like an orchestra. You can point to the string section and say, that's where the violin sound comes from, and you'd be right in a meaningful sense. But the violin sound you actually hear in a symphony is shaped by the acoustics of the hall, by what the other instruments are doing, by the conductor's interpretation, by 1,000 interacting factors. The violin section is necessary for the violin sound, but it's not sufficient on its own. Broca's area is necessary for speech production, But speech production also involves motor cortex controlling your mouth and tongue and vocal cords. And it involves planning areas that help you organize your thoughts before you speak, and it involves emotional circuits that determine your tone of voice, and it involves memory systems that store the words you want to use. Damage Broca's area, and you lose a critical node in that network, and speech production collapses. But that doesn't mean speech production lives in Broca's area the way your car keys live in your pocket. This distinction between a region being necessary for a function and a region being the sole seat of that function is one of the most important ideas in neuroscience, and honestly, it's one that people still get confused about today. When you see those colorful brain scan images in news articles, the ones that light up a specific region and say, "Scientists find the brain area responsible for love," or for jealousy, or for political opinions. Take those with a big grain of salt. What those scans usually show is that a particular region is more active during a particular task. That's useful information, but it doesn't mean that region is the love center or the jealousy center. The brain doesn't work that way. It works through networks, through patterns of activity distributed across many regions, communicating with each other through billions of connections. But we're getting ahead of ourselves. Let's go back to the 19th century, because there's another discovery happening around this same time that's just as important as localization, and it has to do with a question that might seem almost absurdly basic. What is the brain actually made of? What is the physical stuff that does all this thinking and feeling and remembering? And the answer to that question was the subject of one of the most bitter scientific feuds in history. By the mid-1800s, microscopes had gotten good enough that scientists could start looking at brain tissue in real detail. And what they saw was confusing. The brain is incredibly dense with material. If you look at a thin slice of brain tissue under a microscope without any special preparation, it mostly just looks like a tangled, undifferentiated mess. You can't make out individual structures. It's like trying to see individual trees from an airplane flying over a dense forest. Everything just blurs together into a green carpet. So the big question was, is brain tissue actually made up of individual, discrete cells, the way your muscles and your liver and your skin are? Or is it something different, a continuous network, a kind of living web where everything is physically fused together? This might sound like a minor technical question, but it's actually fundamental. Because if the brain is made of individual cells, then those cells must be communicating with each other somehow, sending signals back and forth across tiny gaps. And that means you could potentially understand the brain by understanding those signals. But if the brain is a continuous web, then information might flow through it the way electricity flows through a wire, and the whole picture changes. The man who cracked this problem open, or at least who gave us the tool to crack it open, was a quiet, somewhat melancholy Italian physician named Camillo Golgi, working in the early 1870s in a hospital kitchen in Abbiategrasso, a small town near Milan. And I love this detail because it's such a perfect example of how great science sometimes happens in the most unlikely places. Golgi didn't have a proper laboratory. He was working by candlelight in a kitchen, experimenting with different chemical stains that might make brain tissue more visible under the microscope. And sometime around 1873, he stumbled onto something remarkable. He found that if you hardened brain tissue in potassium dichromate and then soaked it in silver nitrate, something almost magical happened. The silver compound would randomly stain a small percentage of the neurons, maybe 1 or 2%, in their entirety. The cell body, the long branching extensions, everything, rendered in stark black against a clear background. It was like someone had taken that dense forest and randomly illuminated just a handful of trees with spotlights so you could finally see their full shape. Trunk, branches, roots, everything, for the first time. Golgi called it the black reaction, and it was one of the most important technical breakthroughs in the history of neuroscience. For the first time, scientists could see individual brain cells in all their astonishing complexity. And they were complex. These weren't simple little spheres like red blood cells. They had elaborate branching structures extending outward from the cell body, reaching in every direction, some of them stretching remarkable distances. They were beautiful, actually, intricate and delicate, like frost patterns on a window or the branching of a winter tree. But here's the irony. Golgi himself, the man who invented the staining technique that revealed individual neurons in all their glory, looked at his own preparations and concluded that the brain was a continuous network. He believed those branches he was seeing were physically fused together, forming an unbroken web he called a diffuse neural net. And on the other side of this debate was a Spanish scientist named Santiago Ramón y Cajal, who took Golgi's own staining method, refined it, applied it with extraordinary patience and artistic skill, and came to the exact opposite conclusion. Cajal looked at the same kind of preparations and saw individual cells, discrete units separated by tiny gaps, each one a self-contained entity that received signals through its branches and sent signals out through a single long extension called an axon. And what makes this story so wonderfully human, so perfectly illustrative of how science actually works, is that these two men, Golgi and Cajal, shared the Nobel Prize in Physiology or Medicine in 1906 for their work on the structure of the nervous system. They shared the stage in Stockholm, and they used their Nobel lectures to attack each other's theories. Golgi stood up and argued for the continuous network. Cajal stood up and argued for individual cells. At the Nobel ceremony, you can't make this stuff up. Cajal was right, as it turned out. The brain is made of individual cells, neurons, and they communicate across tiny gaps called synapses. And this fact, this seemingly simple anatomical reality, is the foundation of everything we understand about how the brain works. Every thought you've ever had, every memory, every emotion, every dream you're drifting toward right now, all of it comes down to neurons sending signals to each other across synapses. But understanding how those signals actually work, what they're made of, how a thought becomes a chemical, becomes an electrical impulse, becomes a chemical again, that's a whole other story. And it involves some of the most elegant experiments in the history of biology, starting with a frog heart, a German pharmacist's son, and a dream that came to him twice. His name was Otto Loewi, and he was born in Frankfurt, Germany, in 1873. The same year, actually, that Golgi first developed his silver staining technique, which is a nice little coincidence. Loewi trained as a physician, but fell in love with pharmacology, with the way chemicals interact with living tissue, and he spent years studying how the nervous system controls the heart. And here's the thing about Loewi. He had an idea rattling around in his head for years, a hunch, really, about how nerves communicate with the organs they control. The question was simple, but enormous. When a nerve tells your heart to slow down or tells a muscle to contract, what is the actual message? Is it electrical? Is it chemical? Or is it some combination of the two? By the early 20th century, most physiologists assumed the answer was purely electrical. It made intuitive sense. Nerves seem to work like wires. A signal travels down the nerve fiber, hits the target organ, and the organ responds. Clean, fast, electrical. But Loewi suspected something else was going on. He thought that when a nerve signal reached its endpoint, it triggered the release of a chemical substance, and that chemical substance was the actual messenger. The thing that told the heart to slow down, or the muscle to twitch. The nerve didn't zap the organ directly. It released a chemical intermediary. And this idea, if true, would change everything about how we understand the brain. Because it would mean that neural communication isn't just an electrical phenomenon. It's a chemical one too. But he couldn't figure out how to prove it. For years. The idea just sat there, nagging at him. And then, in 1921, something remarkable happened. Lowy later described it himself. He woke up in the middle of the night with the design of an experiment fully formed in his mind, as if it had come to him in a dream. He scribbled notes on a scrap of paper, went back to sleep, and in the morning found that he couldn't read his own handwriting, couldn't remember the idea at all. Gone. Just imagine that frustration, the answer to the question that had consumed you for years, and you can't read your own notes. But the dream came back. The next night, the same idea returned, and this time, Lowy didn't bother writing it down. He got up, went straight to his laboratory in the middle of the night, and performed the experiment right then and there. And it's one of those experiments that's so elegant, so beautifully simple, that it almost makes you laugh. Here's what he did. He took 2 frog hearts, still beating, kept alive in a saline solution, which you can do with frog hearts because they're remarkably resilient. One heart still had its vagus nerve attached. The vagus nerve is this long, wandering nerve. Vagus actually comes from the same Latin root as vagrant and vagabond because it wanders all over the body, and one of its jobs is to slow the heart down. So Lowy stimulated the vagus nerve on the first heart, and sure enough, the heart slowed down. Nothing surprising there. But then he took the saline solution that the first heart had been sitting in, the fluid that had been bathing the heart while the vagus nerve was being stimulated, and he transferred that fluid to the second heart. The second heart, remember, had no vagus nerve attached, no nerve connection at all, and the second heart slowed down too. Think about what that means. The nerve hadn't touched the second heart. No electrical signal had reached it. The only thing that had passed between the 2 hearts was the fluid, the liquid, which meant that when the vagus nerve fired, it released something, some chemical substance, into the fluid, and that substance alone was enough to slow a heart down. Lowy called it Vagusstoff, which is just German for Vegas stuff, which I love because it's so refreshingly unpretentious for one of the most important discoveries in neuroscience. We now know that Vagusstoff was acetylcholine, one of the first neurotransmitters ever identified. And this is where the story of the brain takes a turn that nobody quite expected, because what Lowy proved with his frog hearts applies not just to the connection between nerves and organs, but to the connections between neurons themselves, those synaptic gaps that Kajal had identified. When one neuron sends a signal to another, it doesn't just zap it electrically across the gap. It releases neurotransmitter molecules into the synapse. Those molecules drift across and bind to receptors on the receiving neuron, and that binding is what triggers the next electrical signal. So the process is electrical, then chemical, then electrical again. Electricity runs down the axon, chemistry crosses the gap. Electricity fires in the next cell. It's a relay system, and the handoff point, the synapse, is chemical. Now you might be thinking, okay, that's interesting, But why does it matter whether the signal is electrical or chemical? And the answer is that the chemical step is where almost everything interesting happens. Because a chemical handoff can be modulated. It can be turned up or turned down. The receiving neuron can have more receptors or fewer. The sending neuron can release more neurotransmitter or less. Other chemicals can hang around in the synapse and interfere, or help, or change the message entirely. If neural communication were purely electrical, like current flowing through a wire, there wouldn't be much room for subtlety. But because there's a chemical step, there's an enormous amount of flexibility. And that flexibility is the basis for learning, for mood, for the effects of drugs, for addiction, for mental illness, for basically everything that makes the brain more than just a very complicated circuit board. Lowy shared the Nobel Prize in 1936 with Henry Dale, a British physiologist who had independently done crucial work on acetylcholine and other chemical messengers. And here's a sad footnote to Lowy's story. 2 years after receiving the Nobel, the Nazis annexed Austria, where Lowy was living and working. He was arrested, forced to transfer his Nobel Prize money to a Nazi-controlled bank, and eventually fled to the United States, where he spent the rest of his career at New York University. His dream experiment, one of the most beautiful in the history of biology, performed in a lab he would be forced to abandon at gunpoint. But the knowledge survived, and it opened the floodgates. Because once you know that neurons communicate chemically, the next question is obvious. How many different chemicals are there? Is it just acetylcholine, or are there others? And the answer, as researchers discovered over the following decades, is that there are many. Dozens of neurotransmitters have been identified, each with different roles, different distributions in the brain, different effects on the neurons they touch. You've heard of some of them even if you've never studied neuroscience. Dopamine, serotonin, norepinephrine, GABA, glutamate. These names show up in everything from antidepressant commercials to fitness magazines, and they've become part of everyday language in ways that would have astonished Lowy. Let's talk about a couple of them, because they illustrate just how much the chemical nature of the brain matters for your daily life. Take dopamine. There's a popular idea that dopamine is the pleasure chemical, the thing that makes you feel good when you eat chocolate or get a notification on your phone. And that's not entirely wrong, but it's a massive oversimplification. Dopamine is more accurately described as being involved in motivation, reward prediction, and learning. It's not so much about pleasure itself as it is about the anticipation of pleasure, the wanting rather than the liking, if that makes sense. When something good happens unexpectedly, dopamine neurons fire like crazy. But here's what's really clever. When something good happens that you predicted, the dopamine response is much smaller. And when you predict something good and it doesn't happen, dopamine activity actually drops below baseline. Your brain is essentially running a prediction error system, constantly comparing what it expected to what actually happened, and dopamine is the signal that encodes the difference. This matters enormously for understanding addiction, because addictive substances hijack the system. They cause massive dopamine surges that the brain never predicted, which creates an incredibly strong learning signal. Your brain essentially screams, "Whatever you just did, do it again. That was way better than expected." And over time, the brain adjusts. It downregulates its dopamine receptors, which means you need more of the substance to get the same effect, which is tolerance, and the baseline drops, so without the substance, you feel worse than you did before you ever tried it. The chemistry of the synapse, that little gap that Kajal identified and Lowy proved was chemically mediated, is where addiction lives. Or take serotonin. most of the antidepressant medications prescribed today, the SSRIs, selective serotonin reuptake inhibitors, drugs like fluoxetine, which you might know as Prozac, work by increasing the amount of serotonin available in the synapse. They do this by blocking the reuptake mechanism, the little molecular pumps that normally suck serotonin back into the sending neuron after it's been released. Block those pumps, and serotonin hangs around longer, stimulating the receiving neuron more. And for many people with depression, this helps. But here's an honest admission that neuroscience has to make. We still don't fully understand why it helps. The serotonin hypothesis of depression, the idea that depression is simply caused by low serotonin levels, is almost certainly too simple. SSR is increased serotonin levels within hours, but the therapeutic effects typically take weeks to appear. Something else is happening downstream, something involving changes in receptor sensitivity, neural plasticity, gene expression, and we're still working it out. Which is a good reminder that knowing the parts doesn't mean you understand the whole system. We know about neurotransmitters, we know about synapses, but the brain has roughly 86 billion neurons and something like 100 trillion synaptic connections, and the way those connections interact to produce something as complex as a mood or a personality or a sense of self, that's a level of complexity that still humbles everyone who studies it. And this brings us to something I've been wanting to come back to, the question of how the brain is organized on a larger scale. We talked earlier about Broca and Wernicke, and the idea that specific brain regions handle specific functions. And that's true as far as it goes. But the neurotransmitter story adds a whole new dimension, because neurotransmitter systems don't respect neat anatomical boundaries. Dopamine neurons are clustered in a few small regions deep in the brain, the ventral tegmental area, the substantia nigra, but they project their axons widely, influencing the prefrontal cortex, the limbic system, the basal ganglia. Serotonin neurons are concentrated in the raphe nuclei of the brainstem, but they send fibers to almost every part of the brain. So you have these localized clusters of neurons broadcasting chemical signals across vast networks, modulating how entire brain regions behave. It's like the difference between a spotlight and a mood light. Broca's area is a spotlight, handling a specific task in a specific place, but serotonin is more like the ambient lighting of the whole room, changing the character of everything at once. And this dual organization, specific regions doing specific things, overlaid with broad chemical systems that modulate everything, is part of what makes the brain so hard to understand and so fascinating to study. It also helps explain something that puzzled researchers for a long time, which is how damage to a tiny brain region can sometimes have devastating, highly specific effects, while damage to another region of similar size might produce subtle changes that are hard to even detect. It depends on whether you've knocked out a critical processing node or just slightly altered the tone of a diffuse chemical system. Now, understanding the electrical side of neural signaling, not just the chemical handoff at the synapse, but the electrical impulse that travels down the axon itself, that required its own set of brilliant experiments, and it takes us to a place you might not expect, the coast of England, a laboratory in Plymouth, and a creature with an axon so absurdly large that you can actually see it with the naked eye. The squid giant axon. And when I say giant, I don't mean giant by squid standards. I mean giant by any standard. Most axons in your body are maybe 1 to 20 micrometers across. A micrometer is 1,000th of a millimeter. So we're talking about fibers so thin, you'd need a powerful microscope to even see them. But the long fin inshore squid, Loligopilae, has axons in its mantle that can be up to a millimeter in diameter. That's roughly 100 times thicker than a typical human axon. You could, if you were careful, pick one up with a pair of fine forceps. You could slide a tiny electrode inside it. And that's exactly what two young British physiologists decided to do. Their names were Alan Hodgkin and Andrew Huxley, and the story of what they accomplished is one of the most elegant pieces of scientific detective work in the 20th century. But like so many great stories, it didn't happen all at once, and it almost didn't happen at all. Hodgkin was born in 1914 in Banbury, England. His father died of dysentery during the First World War when Alan was just 3 years old. He grew up fascinated by natural history, birds, insects, the usual trajectory of a curious English boy. But somewhere along the way, he became captivated by the question of how nerves work. By the time he was a student at Trinity College, Cambridge, in the early 1930s, the basic outline was known. Nerves carry electrical signals, and these signals travel as brief pulses called action potentials. But nobody really understood the mechanism. What was actually happening, physically, at the level of the nerve fiber when an impulse passed through. The prevailing theory, championed by a German physiologist named Julius Bernstein back in 1902, was that nerve cells maintain a resting electrical potential across their membrane. The inside of the cell is negatively charged relative to the outside, maybe around negative 70 millivolts or so. Bernstein proposed that when a nerve fires, the membrane briefly becomes permeable to all ions. The voltage difference collapses to zero, and that's the signal. It was a reasonable hypothesis, and it was mostly right, but it was wrong in one crucial detail, and that detail turned out to change everything. Hodgkin began working on nerve impulses in the mid-1930s, first at Cambridge, and then during a fellowship year at the Rockefeller Institute in New York, where he met some of the leading neurophysiologists of the day. He returned to England and teamed up with Andrew Huxley, who was younger, born in 1917, and came from a rather remarkable family. His half-brother was Aldous Huxley, the novelist who wrote Brave New World, and his grandfather was Thomas Henry Huxley, the famous Victorian biologist known as Darwin's bulldog for his fierce public defense of evolution. So Andrew Huxley came from serious intellectual stock, and he turned out to be a formidable experimentalist and mathematician in his own right. In 1939, Hodgkin and Huxley traveled to the Marine Biological Association laboratory in Plymouth, on the southwest coast of England, to work with squid giant axons. The reason they needed those enormous axons was purely practical. The recording technology of the era simply wasn't delicate enough to measure what was happening inside a normal-sized nerve fiber. You couldn't get an electrode inside something 20 micrometers across, not with 1930s equipment. But a squid giant axon, that was big enough to work with. You could thread a fine wire right down the center of it, like sliding a pipe cleaner into a straw, and directly measure the voltage inside the cell while it fired. And here's what they found in that first summer of experiments in 1939. When the nerve impulse passed, the voltage didn't just collapse to zero the way Bernstein had predicted. It actually overshot. The inside of the cell didn't just reach 0 millivolts, it swung positive, up to about plus 40 millivolts, before snapping back. That overshoot was a huge clue. It meant the membrane wasn't just passively leaking ions in both directions. Something active was happening. Something was driving positive charges into the cell, pushing the voltage past zero and into positive territory. And then the war started. Literally. It was the summer of 1939. Hodgkin and Huxley had just made this beautiful observation, and then Hitler invaded Poland, and everything stopped. Hodgkin went off to work on radar for the British military. Huxley worked on gunnery and other military research. Their squid axon experiments were put on hold for 6 years, 6 years. Think about that. You've just glimpsed something extraordinary. You know you're on the edge of understanding one of the fundamental mechanisms of life, and you have to walk away and spend 6 years working on completely different problems while the world tears itself apart. But maybe those years weren't entirely wasted, at least not scientifically. Hodgkin's radar work gave him deep experience with electronics, amplifiers, oscilloscopes, the behavior of electrical circuits, and that expertise would prove invaluable when he returned to neuroscience. Huxley's mathematical skills were sharpened by his wartime work. So when the war ended and they reconvened at Plymouth in 1947 and then again in 1949, they were better equipped than ever. Now, here's where the real genius comes in. They didn't just measure the action potential more precisely. They invented a technique to control it. It's called the voltage clamp, and the basic idea is this. Instead of letting the nerve do whatever it wants and trying to measure the resulting voltage change, you force the membrane to hold at a specific voltage and then measure how much current flows across it at that voltage. You're essentially saying to the nerve, I'm going to pin your voltage right here. Now show me what ions are moving. By systematically stepping through different voltages and recording the currents, they could tease apart exactly which ions were flowing, in which direction, and when. And what they discovered was breathtaking in its elegance. The action potential isn't one event, it's 2 events overlapping in time. First, sodium channels in the membrane open. Sodium ions, which are positively charged and much more concentrated outside the cell than inside, come rushing in through these channels, driven by both the concentration gradient and the electrical gradient. That influx of positive charge is what drives the voltage from negative 70 millivolts up past 0 to about plus 40. That's the overshoot that Bernstein's theory couldn't explain. Then, a fraction of a millisecond later, the sodium channels close and potassium channels open. Potassium ions, which are more concentrated inside the cell, flow outward, carrying positive charge back out and bringing the voltage back down to its resting level. The whole thing takes maybe a millisecond or 2. So the action potential is like a 1-2 punch. Sodium in, potassium out. And the timing is everything. The sodium channels open first and close quickly. The potassium channels open a little later and stay open a bit longer. This precise choreography produces that sharp spike of voltage that constitutes the nerve impulse. And here's what's really clever about the system. It's self-propagating. When one patch of membrane fires, when sodium rushes in and the voltage spikes, that local voltage change is enough to trigger the sodium channels in the adjacent patch of membrane to open, which triggers the next patch, which triggers the next. The impulse rolls down the axon like a wave in a stadium, each section firing in sequence, powered by its own local ion flows. The signal doesn't get weaker as it travels, because each segment generates a fresh burst of current. It's like a line of dominoes, except each domino stands itself back up again after it falls, ready to be knocked down again. Hodgkin and Huxley published their findings in a landmark series of 5 papers in 1952 in the Journal of Physiology. The final paper presented a mathematical model, a set of differential equations, that described exactly how the sodium and potassium currents behaved as functions of voltage and time. And here's the remarkable thing. When they solved those equations, the predicted shape of the action potential matched the real thing almost perfectly. The speed of propagation, the amplitude, the duration, the refractory period, it all fell out of the math. They had essentially reverse-engineered the electrical signaling mechanism of the nervous system from first principles. They shared the Nobel Prize in Physiology or Medicine in 1963, along with John Eccles, who had done important work on synaptic transmission. And the Hodgkin-Huxley model is still taught in every neuroscience program in the world. It's one of those rare achievements that was essentially correct the first time. Later discoveries have added nuance, but the core framework has held up for over 70 years. Now, there's a beautiful detail buried in their work that connects back to something we talked about earlier, the question of speed. Remember, the whole reason the squid has that giant axon is that a bigger axon conducts signals faster. The squid uses those axons to control its jet propulsion escape response. When a predator attacks, it needs to contract its mantle muscles simultaneously and powerfully, and that requires getting the signal from the brain to the muscles as fast as possible. A fatter axon has lower electrical resistance, so the signal propagates faster. It's a brute force solution. Make the cable bigger. But vertebrates, animals with backbones, including you, evolved a much more elegant solution to the same problem. Instead of making axons enormous, they wrapped them in insulation. That insulation is called myelin, and it's made by specialized glial cells, Schwann cells in the peripheral nervous system, oligodendrocytes in the brain, and spinal cord. These cells wrap around the axon in tight spirals, like electrical tape around a wire, creating a fatty sheath that prevents ions from leaking out through the membrane. But here's the trick. The myelin doesn't cover the entire axon. It leaves tiny gaps, about 1 micrometer wide, spaced roughly a millimeter apart. These gaps are called nodes of Ranvier, named after the French anatomist Louis-Antoine Ranvier, who described them in 1878. And the action potential doesn't travel smoothly down a myelinated axon the way it does in the squid's giant axon. Instead, it jumps from node to node. The electrical signal races passively through the insulated segments, very fast, because there's no ion exchange happening, just pure electrical conduction, and then regenerates at each node where the sodium channels are densely packed. This jumping conduction is called saltatory conduction, from the Latin word saltare, meaning to leap or dance, and it's dramatically faster. A squid giant axon, despite being a millimeter across, conducts at about 25 meters per second. A myelinated human motor neuron, maybe 20 micrometers in diameter, 50 times thinner, can conduct at over 100 meters per second, 4 times faster at a 50th of the size. That's an extraordinary engineering advantage. It's why you can have a brain with billions of neurons packed into a space the size of a cantaloupe, all communicating at high speed instead of needing axons as thick as spaghetti noodles. And when myelin breaks down, you see just how critical it is. Multiple sclerosis is a disease in which the immune system attacks the myelin sheaths in the brain and spinal cord. As the insulation degrades, signals slow down, become unreliable, or fail to propagate at all. The symptoms depend on which axons are affected. Vision problems if the optic nerve is hit, muscle weakness if motor pathways are damaged, numbness if sensory fibers lose their coating. It's a devastating illustration of how much the brain's function depends not just on the neurons themselves, but on the infrastructure that supports them. And that brings up something that I think doesn't get enough attention when people talk about the brain, which is that neurons are actually a minority of the cells in there. For a long time, you'd hear the claim that glial cells outnumber neurons 10 to 1. That turns out to be an overestimate. More recent counting methods, pioneered by the Brazilian neuroscientist Susana Herculano Hausel in the 2000s, suggest the ratio is closer to 1 to 1. Roughly 86 billion neurons and about the same number of glial cells. But even at a 1 to 1 ratio, that's an enormous population of non-neuronal cells, and for most of the history of neuroscience, they were almost completely ignored. The word glia comes from the Greek for glue. They were literally thought of as just the packing material that held the important cells in place, but it turns out they're doing far more than anyone suspected. Take astrocytes, for instance. These are star-shaped glial cells. The name literally means star cell, and they're everywhere in your brain. Each astrocyte extends thousands of fine little processes, these delicate branching arms, and those arms wrap around synapses. One single astrocyte can be in contact with hundreds of thousands of synapses simultaneously. For a long time, people assumed they were just providing structural support, maybe helping clean up waste products. But starting in the 1990s, researchers began to realize that astrocytes are actually listening to the conversations happening at those synapses. They detect neurotransmitters being released, and they respond, sometimes by releasing their own signaling molecules, sometimes by adjusting how much neurotransmitter gets cleared from the synaptic cleft, sometimes by changing the local blood flow to deliver more oxygen and glucose to neurons that are working hard. That last one is worth pausing on because it connects to something you've probably heard of. When someone gets a brain scan, an fMRI, a functional magnetic resonance imaging scan, what the machine is actually measuring isn't neural activity directly. It's measuring changes in blood flow. The assumption is that when a brain region becomes more active, it needs more blood, so increased blood flow is a proxy for increased neural activity. And that assumption works pretty well in practice. But here's the thing. The cells that are largely responsible for regulating that local blood flow are astrocytes. So in a very real sense, every colorful brain scan you've ever seen in a news article about psychology or neuroscience is actually showing you astrocyte activity as much as it's showing you neuron activity. The two are deeply intertwined, and astrocytes do something else that's genuinely remarkable. They're connected to each other through gap junctions, direct physical channels between adjacent cells, forming vast networks that span large regions of the brain. Through these networks, they can pass ions and small molecules to each other, creating slow waves of calcium signaling that wash across the astrocyte network on a completely different time scale than the fast electrical spikes of neurons. Some researchers have proposed that these calcium waves might represent a kind of parallel processing system, a slow background layer of computation happening alongside the fast neural signaling. That's still debated, and we should be honest about that. It's one of those ideas that's tantalizing, but not fully proven. But even the conservative view of astrocytes, the stuff that's well established, makes them far more than glue. They regulate synaptic strength. They influence which connections get maintained and which get pruned. They help form the blood-brain barrier, and they respond to injury by forming scar tissue. Their infrastructure, yes, but they're active, dynamic, responsive infrastructure. Then there are oligodendrocytes, which we've already met. Those are the cells that produce myelin in the central nervous system. And there are microglia, which are the brain's resident immune cells. Microglia are actually fascinating in their own right, because they're not really brain cells at all, developmentally speaking. They originate from the same lineage as the macrophages in the rest of your body. They're essentially immune cells that migrated into the brain early in development and took up permanent residence. They patrol the brain constantly, extending and retracting their processes, sampling the environment, looking for signs of damage or infection. When they find something wrong, they can engulf debris, release inflammatory signals, and recruit other immune responses. But in the healthy brain, they also play a role in synaptic pruning. They literally eat synapses. During development, and even in the adult brain, microglia selectively eliminate certain synaptic connections, and this pruning turns out to be essential for normal brain function. There's growing evidence that when microglial pruning goes wrong, when it's too aggressive or not aggressive enough or targeted incorrectly, it may contribute to neurodevelopmental conditions and neurodegenerative diseases. Some researchers think excessive microglial pruning might play a role in schizophrenia, where there seems to be an abnormal loss of synaptic connections during adolescence and early adulthood. So the picture of the brain that's emerging in the 21st century is far richer and messier than the one that existed even 30 years ago. It's not just neurons firing in circuits. It's neurons embedded in a dense ecosystem of supporting cells, all influencing each other, all contributing to the computation in ways we're still working out. And that word, computation, is worth thinking about because it brings us to one of the most consequential ideas in the history of brain science, which is the question of whether the brain is, in some meaningful sense, a computer. Now, this analogy has a complicated history, and it's one of those things where the metaphor has been both incredibly useful and genuinely misleading, depending on how literally you take it. The idea didn't come out of nowhere. It emerged from a very specific moment in the middle of the 20th century, when several different intellectual threads converged in a way that felt almost electric. You had Alan Turing working in the 1930s on the theoretical foundations of computation, defining what it meant for a machine to be able to calculate anything that was calculable. You had Claude Shannon at Bell Labs in 1937, showing that electrical switching circuits could implement Boolean logic, that you could build physical machines that performed logical operations. You had Warren McCulloch and Walter Pitts publishing their landmark 1943 paper, proposing that neurons could be understood as logical units, that a network of simplified neurons could, in principle, compute any function that a Turing machine could compute. And then you had the actual construction of electronic computers during and after the Second World War. Machines like ENIAC and the ones that came after, which made the abstract idea of computation into a tangible, physical reality. The McCulloch-Pitts paper is especially interesting because it really was the spark that launched the whole field of computational neuroscience. McCulloch was a neurophysiologist and philosopher, a genuinely unusual character, the kind of person who could quote Leibniz and dissect a cat brain in the same afternoon. Pitts was a self-taught mathematical prodigy, a teenager from Detroit who had essentially educated himself by reading in the public library. The story goes that Pitts, at age 12, read Bertrand Russell's Principia Mathematica and wrote Russell a letter pointing out what he thought were errors. Russell was apparently impressed enough to invite Pitts to come study with him in Cambridge, though Pitts never went. Instead, he ended up in Chicago, where he met McCulloch, and the two of them developed this model of the neuron as a binary threshold unit, either firing or not firing, either on or off, just like a switch in a logic circuit. If the total input to the neuron exceeded a threshold, it fired. If not, it didn't. Simple as that. And here's what was so powerful about this idea. McCulloch and Pitts showed that networks of these simplified neurons could perform any logical operation and, or not, all the basic gates, which meant, in principle, that the brain could do anything a computer could do, because any computation can be built from those basic logical operations. It was a beautiful, elegant framework, and it electrified people. It suggested that understanding the brain was, at some level, an engineering problem. Figure out the circuit, figure out the logic, and you'd understand the mind. The person who really ran with this was John von Neumann, the Hungarian-American mathematician, who was arguably the most brilliant polymath of the 20th century. Von Neumann was involved in the design of some of the earliest stored program computers, and he explicitly drew on the neuron analogy. In his architecture for digital computers, the von Neumann architecture that still underlies most computers today, he used language borrowed directly from neuroscience. The processing units in his designs were sometimes described in terms that echoed McCulloch and Pitz's neural networks. And late in his life, von Neumann wrote a book called The Computer and the Brain, published posthumously in 1958, in which he tried to seriously compare the two systems. He was honest about the differences. He noted that the brain was far slower than a computer in terms of individual switching speed, but massively more parallel, with billions of operations happening simultaneously rather than one at a time. He noted that the brain seemed to use a different kind of logic than digital computers, something more statistical, more probabilistic, more tolerant of noise and error. And that last observation is really the crux of why the computer metaphor, while useful, can lead you astray if you grip it too tightly. A digital computer is precise. Every bit is either a 0 or a 1. Every operation follows exact rules, and if a single transistor gives the wrong answer, the whole computation can fail. The brain is nothing like that. Neurons are noisy. They don't fire at exactly the same threshold every time. Synaptic transmission is probabilistic. When an action potential arrives at a synapse, there's only a certain probability that a vesicle of neurotransmitter will actually be released. The same stimulus presented twice will produce slightly different patterns of neural activity each time. And yet, the brain manages to produce remarkably reliable behavior out of all this noise. You can catch a ball thrown to you. You can recognize your mother's face in a crowd. You can understand a sentence spoken in a noisy restaurant. The brain achieves reliability not through precision of individual components, but through redundancy, through averaging over large populations of neurons, through architectures that are inherently fault-tolerant. Think about what that means from an engineering perspective. It's a completely different design philosophy than what we use in conventional computers. It's more like, imagine building a bridge not out of a few perfectly machined steel beams, but out of millions of slightly imperfect fibers, each one a little different, each one carrying a slightly different share of the load, but collectively forming a structure that's incredibly robust. If a few fibers break, the bridge still holds. If a whole section degrades, the remaining fibers redistribute the load. That's closer to how the brain works, and it's why brain damage doesn't usually cause the kind of catastrophic, total failure you'd see if you pulled a chip out of your laptop. Instead, brain damage tends to cause partial graded deficits, a little worse at remembering names, a little slower at processing visual motion, a slight change in personality. The system degrades gracefully, as engineers like to say. This robustness is also why the brain can do something that conventional computers have historically been terrible at, which is pattern recognition in messy, real-world conditions. For decades, computers could beat any human at arithmetic, but couldn't reliably tell the difference between a picture of a cat and a picture of a dog. That always struck people as paradoxical. How could a machine that calculates 1,000,000 times faster than you not be able to do something a toddler finds trivial? The answer is that recognizing a cat isn't really a computation problem in the way that adding numbers is. It's a pattern matching problem. And the brain has evolved architectures that are spectacularly good at pattern matching. Architectures that exploit exactly the kind of noisy, parallel statistical processing that von Neumann noticed back in the 50s. And this is where the story takes a turn that connects directly to the world you're living in right now. Because the modern revolution in artificial intelligence, the deep learning systems that can now recognize faces, translate languages, generate text, beat humans at games, these systems are, in a loose but meaningful sense, descendants of that 1943 McCulloch-Pitts paper. They're built from artificial neural networks, layers of simplified neuron-like units connected by adjustable weights, learning from data rather than being explicitly programmed. The connection to real neuroscience is complicated. Modern AI researchers will be the first to tell you that their networks are inspired by the brain, but don't really work like the brain in most of the details. But the core insight that you can build intelligent behavior out of large networks of simple, noisy, threshold-like units, that insight traces straight back to McCulloch and Pitts sitting in Chicago in the early 1940s, trying to figure out how a neuron could be modeled as a logical device. But here's what's really interesting to me about all of this, and it's something that often gets lost in the excitement about artificial intelligence and computational models. The brain isn't just a pattern recognizer. It isn't just a computer of any kind, not really. It's also the thing that generates your conscious experience, the felt quality of seeing red, of tasting coffee, of feeling anxious before a job interview. And that problem, the problem of consciousness, of how physical matter gives rise to subjective experience, is something that no amount of engineering brilliance has yet been able to crack. And honestly, it might be the deepest question human beings have ever asked about themselves. Let's sit with this for a moment, because it deserves some real space. You know what it feels like to be you. Right now, lying there, you have a felt sense of existing. The weight of your body against the mattress, the texture of the air in the room, maybe a faint hum from somewhere. These aren't just data points being processed, they're experienced. There's something it's like to be you right now, in a way that there presumably isn't something it's like to be your thermostat, even though your thermostat also takes in information and responds to it. And the question, the really hard question, is why? Why does all that electrochemical activity in your skull, all those action potentials firing and neurotransmitters binding and glial cells doing their calcium wave thing, why does any of that produce the inner movie of conscious experience? Why isn't it all just happening in the dark, so to speak? The philosopher David Chalmers gave this a name in 1995 that stuck. He called it the hard problem of consciousness. And what's clever about that framing is the implicit contrast with what he called the easy problems, which, to be clear, aren't easy at all. The easy problems are things like explaining how the brain integrates information, how it directs attention, how it distinguishes between being awake and being asleep, how it controls behavior. Those are fantastically difficult neuroscience problems, but they're the kind of problems where you can imagine, in principle, that a sufficiently detailed mechanistic account would give you a satisfying answer. You trace the circuits, you identify the computations, you explain the behavior. Done. The hard problem is different because even if you had a complete wiring diagram of the brain, every synapse mapped, every firing pattern recorded, every chemical interaction cataloged, you'd still face this explanatory gap. Why does all of that machinery feel like anything from the inside? Now, not everyone agrees that the hard problem is a real problem. Some very smart people, Daniel Dennett was probably the most famous, argued that consciousness isn't some extra magical ingredient sitting on top of brain processing. It is the processing. When you've explained all the functional stuff, all the discrimination and integration and reporting and attention, you've explained consciousness. There's no residual mystery. The feeling that there's a hard problem, Dennett would say, is itself a kind of cognitive illusion, a failure to appreciate just how much work the functional explanation is doing. And you know what? There's something bracing about that position. It refuses to be mystified. But most people, including most philosophers and a lot of neuroscientists, find it unsatisfying. It feels like it's explaining away the phenomenon rather than explaining it. So where does neuroscience actually stand on this? Well, here's the honest answer. We don't have a solution, but we do have some fascinating frameworks that try to bridge the gap, and they've generated real experimental predictions, which is more than you might expect from something this philosophical. One of the most influential is called integrated information theory, usually just called IIT, developed primarily by Giulio Tononi, a neuroscientist and psychiatrist at the University of Wisconsin-Madison. Tononi started from the subjective side of things, which is unusual. He asked, what are the essential properties of conscious experience itself? And he identified two big ones. First, every conscious experience is informative. It's this particular experience and not any of the countless other experiences it could have been. When you see a red circle, that experience carries information, precisely because it rules out all the other things you could be experiencing. Second, every conscious experience is integrated. It's unified. It comes as a whole. You don't experience the redness separately from the circularity, separately from the location in your visual field. It's all one seamless experience. From these starting points, Tononi built a mathematical framework. He proposed a quantity he called phi, which measures how much integrated information a system generates, essentially, how much the system as a whole is more than the sum of its parts. And his claim is striking. Consciousness is identical to integrated information. A system is conscious to the degree that it has high phi. This means consciousness isn't something the brain does. It's something the brain is by virtue of its particular causal architecture, the way its parts are wired together, such that they're both highly differentiated and highly integrated. Here's what's wild about this. IIT makes some predictions that are genuinely testable and some that are genuinely weird. On the testable side, it predicts that the cerebellum, that dense structure at the back of your brain with more neurons than the rest of the brain combined, should contribute very little to consciousness because its internal architecture is highly modular and repetitive, meaning low integration, meaning low phi. And that actually lines up with clinical evidence. People with cerebellar damage have motor problems, coordination issues, but they don't lose consciousness. Meanwhile, the cerebral cortex, with its massively interconnected, recurrent wiring, would have high phi, and damage there does affect conscious experience profoundly. On the weird side, IIT implies that consciousness isn't exclusive to biological brains. Any system with the right causal structure and sufficiently high phi would be conscious, which could include, in principle, certain kinds of artificial systems, but probably not the digital computers we currently build, because their architecture, despite being able to simulate anything, has low intrinsic integration. A simulated brain, running on a conventional computer, would not be conscious according to IIT, even if it behaved identically to a real brain. That's a strange and controversial claim. There's another major framework that takes a completely different approach. It's called global workspace theory, originally proposed by the cognitive scientist Bernard Baars in the late 1980s and later developed into a more neuroscientifically grounded version by Stanislas Dehaene and Jean-Pierre Changeux in France. The metaphor here is theatrical. Imagine a stage in a dark theater. Many things are happening backstage. Unconscious processing, competing neural signals, all sorts of computations running in parallel. But only a few of these make it onto the brightly lit stage, where they become available to a wide audience of cognitive processes: memory, language, decision-making, motor planning. That moment of broadcast, when information goes from being locally processed to being globally available across the brain, that's what consciousness is, according to this theory. Dehaene and his colleagues have done beautiful experiments supporting this. They use a technique called masking, where they flash a word on a screen very briefly, and then immediately cover it with another image. If the flash is short enough, people genuinely don't see the word. It's processed unconsciously. But if you make the flash just a tiny bit longer, crossing a threshold of about 50 milliseconds, something dramatic happens in the brain. There's a sudden, late wave of neural activity, about 300 milliseconds after the stimulus, that sweeps across the prefrontal and parietal cortex. Dehaene calls this the ignition, a nonlinear, all-or-nothing event where the information suddenly becomes globally available. Below the threshold, local processing only. Above it, global broadcast and conscious awareness. It's remarkably clean as far as neural signatures go. Now, these two theories, IIT and global workspace, actually make different predictions in certain cases, which means in principle you could design experiments to distinguish between them. There's actually a large-scale collaborative project called the Adversarial Collaboration on Consciousness that's been doing exactly this, with Tononi and Dehaene's groups agreeing in advance on experiments whose results would favor one theory over the other. The early results, published around 2023, were mixed. Some findings fit one theory better, some the other. Which is kind of what you'd expect with something this hard. But the fact that consciousness research has reached the point of adversarial collaborations with pre-registered predictions, that's a sign of how much the field has matured. This isn't armchair philosophy anymore. It's empirical science, even if the question it's grappling with is the most philosophical question there is. And there's something else worth noticing here, something that connects back to everything we've been talking about. Both of these theories rely heavily on the kind of brain architecture we've already explored: the recurrent connections, the long-range fibers linking distant cortical areas, the way information doesn't just flow forward from sensation to action, but loops back and forth, getting integrated, refined, compared with predictions. Remember how we talked about the brain's parallel, distributed processing? How von Neumann noticed that the brain achieves reliability through redundancy rather than precision? Consciousness theories are telling us that this architecture isn't just a workaround for noisy hardware. It might be the very thing that makes experience possible. The integration, the recurrence, the massive interconnectedness. These aren't bugs or compromises. They might be the whole point. There's another dimension to consciousness that neuroscience has gotten much better at studying, and it's something you experience every single night. sleep itself. The fact that you lose consciousness when you fall into deep sleep and regain a strange, hallucinatory version of it when you dream. This is a huge clue, and researchers have been mining it for decades. Here's something remarkable that Tononi's group discovered. When you're in deep, dreamless sleep, the slow-wave sleep that dominates the early part of the night, your cortical neurons are still active. They're not shut off. They're actually firing in these big, synchronized waves, all going up and down together. But here's the key. The long-range connectivity between different brain areas drops dramatically. You can demonstrate this with a technique called TMSEEG, where you zap a spot on the cortex with a magnetic pulse and watch how the resulting activity spreads. When you're awake, that pulse sends ripples of complex, differentiated activity cascading across the brain. It echoes and reverberates in intricate patterns. When you're in deep sleep, the same pulse produces a simple wave that dies out locally. The brain is active but disconnected. The parts aren't talking to each other. And according to IIT, that's exactly why consciousness fades. Not because the neurons stop firing, but because integration collapses. Dreaming is the fascinating counterpart. During REM sleep, the brain's internal connectivity partially restores. Long-range communication comes back, cortical activity becomes complex and differentiated again, and, not coincidentally, you become conscious. You have experiences, vivid, bizarre, emotionally charged experiences. But the content is generated internally rather than driven by sensory input, because the thalamic gates that relay sensory information are largely shut. You're conscious, but you're conscious of a world your brain is constructing entirely from the inside. Which, if you think about it, raises the unsettling possibility that this is what your brain is always doing, constructing a world from the inside. And waking perception is just the version that's constrained and corrected by incoming sensory data. This idea, that perception, is fundamentally a constructive process, that your brain is essentially dreaming all the time, and using sensory input to keep the dream on track, has become one of the most exciting frameworks in modern neuroscience. It goes by several names: predictive processing, predictive coding, the Bayesian brain, and its roots go back surprisingly far, at least to Hermann von Helmholtz in the 1860s, who called perception unconscious inference. But the modern version has been developed and championed by people like Carl Friston at University College London, whose free energy principle attempts to unify perception, action, learning, and even consciousness under a single mathematical framework. The core idea is deceptively simple. Your brain doesn't passively receive sensory information and build a picture of the world from the bottom up. Instead, it's constantly generating predictions, top-down models of what it expects to encounter, and then comparing those predictions against the actual sensory signals coming in. What gets passed up through the hierarchy isn't the raw sensory data itself, but the prediction errors, the mismatches between what the brain expected and what it got. Perception, in this view, is the brain's best guess about the causes of its sensory inputs, updated by surprise. Think about what that means for something as simple as walking into your kitchen. Your brain has a detailed model of that kitchen, where the counter is, what color the walls are, where the light switch sits. Most of the sensory input confirms the prediction, so most of it gets explained away and never reaches conscious awareness. You don't consciously perceive every familiar detail. But if something is different, if someone moved the furniture, or there's an unexpected smell, the prediction error spikes, attention gets allocated, and you become consciously aware of the discrepancy. Your brain notices what it didn't predict. This is why you can drive a familiar route and arrive with almost no memory of the journey. Your predictions were so good that there was almost nothing left to consciously process. And this framework explains some genuinely puzzling phenomena. Take visual illusions. The reason your brain can be so confidently wrong about what it's seeing is that perception was never a direct readout of reality in the first place. It's an inference, a best guess, and sometimes the priors, the brain's expectations, override the actual data. Or think about phantom limb pain, where people who've lost an arm still feel agonizing pain in fingers that no longer exist. The brain's predictive model still includes that arm. It's still generating predictions about sensory input from a limb that can't send any signals back, and the resulting prediction errors get interpreted as pain. The model hasn't been updated to match the new reality of the body. Friston's version of all this, the free energy principle, takes the predictive framework and wraps it in a formalism borrowed from statistical physics and machine learning. The idea is that any self-organizing system that persists over time, any system that resists the tendency toward disorder, must minimize something called variational free energy, which you can think of as a measure of surprise, or more precisely, the difference between the system's internal model and the actual state of the world. The brain minimizes free energy in two ways. It updates its internal model to better predict sensory input, which is perception and learning, or it acts on the world to make the world match its predictions, which is action. You don't just passively model the world, you actively shape it to conform to your expectations. When you're cold, you don't just update your model to include the information, "I am cold." You put on a sweater. You change the world to match the prediction that you should be comfortable. Now, Friston's framework is mathematically dense and genuinely controversial. Some people think it's the closest thing neuroscience has to a grand unified theory. Others think it's so general that it explains everything and therefore explains nothing, that any system can be described as minimizing free energy if you squint hard enough. The debate is ongoing and lively. But regardless of where you fall on Friston, the broader predictive processing framework has been enormously productive. It's generated testable predictions. It's influenced clinical research on conditions like autism and schizophrenia, where the balance between predictions and prediction errors may be disrupted. And it's given us a much richer picture of what the brain is actually doing moment to moment. And it connects to something deeply relevant to the experience you're having right now as you lie there, possibly drifting toward sleep. Because one of the things your brain has to predict is its own internal state. Your brain doesn't just predict what's happening out there in the world, the sounds in the room, the temperature of the air, the feel of the pillow against your cheek. It also predicts what's happening in here, inside your body. Your heart rate, your breathing, the tension in your muscles, your level of arousal. And as you start to fall asleep, what's happening is that your brain is gradually shifting its predictions. It's predicting less sensory engagement, less motor readiness, less alertness. And because prediction drives action, remember, you don't just model the world, you shape it to match your expectations. Your brain's prediction that you are falling asleep actually helps make it happen. Your muscles relax because your brain expects them to relax. Your breathing slows because your brain models slower breathing. Sleep, in a very real sense, is a self-fulfilling prophecy. Your brain predicts it, and the body follows. And that's a beautiful place to sit for a moment, because it connects so many things we've been talking about. The predictive brain isn't some abstract theoretical construct. It's the thing that's carrying you towards sleep right now, adjusting your physiology in real time, dampening sensory signals, quieting the motor system, shifting the whole organism into a different mode of operation. And the fact that it can do this, the fact that your brain can predict and regulate its own internal states, brings us to something that neuroscience has only recently started taking seriously, something called interoception. You've heard of the five senses, right? Sight, hearing, touch, taste, smell. That's the list we all learn as kids, but it's wildly incomplete. You also have proprioception, your sense of where your body is in space, the thing that lets you touch your nose with your eyes closed. You have vestibular sense, your sense of balance, anchored in the inner ear. And then there's interoception, which is your sense of what's happening inside your body. Your heartbeat, your gut, your lungs, your blood sugar, your temperature, the subtle signals of inflammation or fatigue or hunger. For a long time, interoception was treated as a kind of background process, something the autonomic nervous system handled on its own without bothering the conscious brain. But starting in the late 1990s and accelerating through the 2000s and 2010s, researchers began to realize that interoception isn't just plumbing. It's fundamental to how you experience emotions, how you make decisions, and even how you construct your sense of self. The key figure here is a neuroscientist named Bud Craig, A.D. Craig, at the Barrow Neurological Institute in Phoenix. In a series of influential papers starting around 2002, Craig mapped out the interoceptive pathway in remarkable detail. He showed that signals from inside the body, from the viscera, the skin, the muscles, the blood vessels, travel along small diameter nerve fibers to a specific part of the spinal cord, then up to a brain region called the posterior insula, and finally forward to the anterior insula, which is tucked deep in the folds of the cortex, hidden beneath the temporal lobe. And here's what Craig argued was happening at each step. The brain was building a progressively more refined map of the body's internal state. By the time the signal reaches the anterior insula, it's not just raw data about heart rate or gut motility. It's an integrated representation of how your body feels right now. And Craig proposed that this integrated interoceptive map, is the neural basis of subjective feeling. Not just emotion in the dramatic sense, but the basic felt quality of being alive, the background hum of existence. The thing that makes this moment feel like something rather than nothing. Now think about what that means in the context of everything we've been exploring. Remember the hard problem of consciousness, the question of why there's something it's like to be you. Craig's work doesn't solve the hard problem. Nobody's solved it, but it gives us a much more concrete place to look. The anterior insula lights up in brain imaging studies whenever people are asked to attend to their own feelings, their own heartbeat, their own sense of self. Damage to the insula can produce bizarre disruptions in self-awareness, and the interoceptive system is deeply connected to the emotional brain, the amygdala, the hypothalamus, the prefrontal cortex, in ways that suggest emotion isn't something that happens to you after you perceive the world. It's woven into perception itself. This actually echoes a very old debate in psychology, one that goes back to the 1880s. William James, the great American psychologist, brother of the novelist Henry James, proposed something counterintuitive in 1884. He said that emotions aren't caused by your perception of a situation. Instead, emotions are your perception of your body's response to a situation. You don't see a bear, feel afraid, and then run. You see a bear, your body starts running, your heart pounds, your muscles tense, and then you feel afraid. Because fear is just your brain's interpretation of all those bodily changes. The Danish physiologist Carl Lang proposed something very similar around the same time, so it became known as the James Lang theory. And people argued about it for over a century. It seemed too simple. Surely emotions start in the brain, not the body, right? Well, the modern interoceptive view suggests James was more right than wrong, though the picture is more nuanced than he could have known. The neuroscientist Lisa Feldman Barrett, working at Northeastern University, has developed what she calls the theory of constructed emotion, which builds on both interoception and predictive processing. Barrett argues that emotions aren't hardwired circuits in the brain. There's no single fear center or anger center, despite what you might have heard about the amygdala. Instead, the brain constructs emotional experiences on the fly by combining interoceptive signals from the body with predictions based on past experience and current context. Your brain is constantly asking, in effect, given what's happening in my body right now and what's happening in the world around me, what is this feeling most likely to be? And it answers that question by drawing on a lifetime of learned categories that are shaped by culture, language, and personal history. This is why the same bodily state can feel like excitement in one context and anxiety in another. Your heart is pounding. Your palms are sweating. Are you about to give a speech? Or are you on a roller coaster? The raw interoceptive data might be nearly identical, but the emotion you experience is different because your brain's prediction is different. The context shapes the construction. And Barrett has shown that people who have richer emotional vocabularies, people who can distinguish between, say, irritation and frustration and resentment, rather than just lumping everything under angry, actually have better emotional regulation and better mental health outcomes. The granularity of your concepts shapes the granularity of your experience. Language literally changes what you feel. So here's where all of this converges into something really profound about the brain as a whole. You've got the predictive processing framework, saying the brain is fundamentally a prediction machine, constantly generating models of the world and updating them based on error signals. You've got the interoceptive system saying the brain is also predicting and modeling its own internal body states, and that this is the basis of feeling and emotion. You've got constructed emotion theory saying that even something as seemingly basic as fear or joy is actually an active construction, not a passive readout. And you've got the consciousness theories we talked about earlier, integrated information theory and global workspace theory, trying to explain why any of this feels like anything at all. These aren't competing stories. They're different angles on the same astonishing fact. Your brain is building your entire experienced reality from the inside out, moment by moment, using prediction, memory, and a constant stream of signals from both the world and your own body. And this brings us to something deeply practical, something that matters for medicine and for understanding the people around you. Because if the brain is constructing experience, including the experience of pain, of emotion, of bodily sensation, then disruptions in that construction process can produce real suffering that doesn't show up on any conventional medical test. Think about chronic pain conditions where there's no clear tissue damage. Think about functional neurological disorders where people experience genuine paralysis or seizures without any detectable structural problem in the nervous system. For a long time, these conditions were dismissed. It's all in your head, doctors would say, which was both technically true and deeply unhelpful. But the predictive processing framework gives us a much better way to understand what's going on. If the brain's predictions about the body become miscalibrated, if the brain starts predicting pain where there is no injury, or predicting paralysis where the motor pathways are intact, then the person genuinely experiences that pain or that paralysis. It's not imaginary. It's not faked. It's a prediction error in the deepest sense, a glitch in the brain's model of its own body. And understanding it that way opens up real treatment possibilities, because you can work on recalibrating the predictions rather than searching fruitlessly for damage that isn't there. Carl Friston's free energy framework even offers a formal way to think about mental illness more broadly. Anxiety, in this view, is what happens when the brain assigns too much weight to prediction errors, when every unexpected signal gets treated as a threat, when the system is perpetually surprised by the world and can't settle into a stable model. Depression might involve the opposite problem, in some respects. A brain that's become too rigid in its predictions, too convinced that nothing will change, too resistant to updating its model in response to new positive information. These are simplifications, of course. Mental illness is complex, multifactorial, shaped by genetics and environment and social context and sheer bad luck. But the predictive framework gives clinicians and researchers a common language for thinking about very different conditions. And it's already influencing how therapy and medication are understood. And here's something else worth sitting with. All of this, the prediction, the construction, the modeling, it's happening right now, in you, as you listen to these words. Your brain is predicting what I'm going to say next. It's modeling the meaning of each sentence before I finish it. It's regulating your body based on its prediction that you are safe, that you are comfortable, that sleep is approaching. And every now and then, if I say something unexpected, there's a tiny flicker of prediction error, a moment where your brain updates its model, incorporates the new information, and settles back into its rhythm. That process, that constant cycle of prediction and correction, is what it feels like to be conscious. Or at least, that's one way to think about it. But there's one more frontier we haven't touched yet, and it's the one that's reshaping neuroscience as we speak. Because everything we've discussed, from Broca's area to neurotransmitters, to glial cells, to predictive processing, has been about understanding the brain as it naturally exists. But what happens when you start to change it? Not just with drugs, which we've touched on, but with technology. What happens when you start reading the brain's signals from outside, or writing new signals in? What happens when the boundary between the brain and the machine starts to blur? That's the question that takes everything we've been talking about tonight and pushes it right to the edge of what it means to be human. And the story starts, as so many of these stories do, with someone trying to solve a very specific problem. In the early 1990s, a neuroscientist named John Donahue at Brown University was studying how the motor cortex controls movement. He wasn't trying to build a mind-reading machine. He was just trying to understand the basic code. How does a population of neurons in your brain translate the thought, "reach for that cup of coffee," into a precise sequence of muscle activations that moves your arm, opens your hand, closes your fingers around the handle, and lifts? And what he found, building on decades of work by people like Apostolos Georgopoulos, was that individual motor neurons don't encode specific muscles. Instead, each neuron has a preferred direction. It fires most vigorously when you're reaching in a particular direction, and less vigorously for other directions. and the actual movement emerges from the combined activity of a whole population of these neurons, each one casting a kind of vote about which direction to go. The brain's movement command is a population code, a collective signal that you can read if you know how to listen. Now, here's where it gets really interesting. Donahue and his colleagues realized that if you could record from enough of these neurons simultaneously, you could decode the intended movement in real time. Not after the fact, not by analyzing data overnight, but right now, as the person thinks about moving. And the reason this mattered so profoundly is that there are people in the world who have perfectly functioning motor cortices but can't move. People with spinal cord injuries, where the connection between brain and body has been severed. People with ALS, where the motor neurons in the spinal cord and brainstem are dying, but the brain itself is still generating movement commands into the void. What if you could intercept those commands and route them somewhere useful? To a computer cursor, to a robotic arm, to the person's own paralyzed limb through electrical stimulation? This was the vision behind BrainGate, the brain-computer interface project that Donahue launched in collaboration with a biomedical engineer named Abby Kaplan, and later with neurosurgeon Lee Hochberg. The core technology was a tiny array of electrodes, a device called the Utah array, about the size of a baby aspirin, with 96 hair-thin silicon electrodes that could be implanted directly into the motor cortex. Each electrode could pick up the electrical activity of one or a few nearby neurons. And in 2004, they implanted one of these arrays in a man named Matthew Nagel. Nagel was 25 years old and had been paralyzed from the neck down after being stabbed in the neck 3 years earlier. He couldn't move his arms or legs. But when the BrainGate system was turned on, and he simply thought about moving his hand, the neural signals were decoded in real time, and he was able to move a cursor on a computer screen. He could open simulated email. He could control a television. He could even open and close a prosthetic hand, just by thinking about it. Think about what that means for a second. The motor cortex was still generating the same population codes it always had, still casting those directional votes, still sending commands down pathways that no longer connected to anything. And the machine was listening, translating those commands into actions in the world. The boundary between Matthew Nagel's brain and the computer wasn't a wall anymore. It was a bridge. Now, BrainGate was remarkable, but it was also crude by today's standards. The Utah array records from a tiny patch of cortex. The signals degrade over months as scar tissue forms around the electrodes. The system required a physical connector protruding from the skull, which created infection risks. And the decoding algorithms, while impressive, were relatively simple, but it proved the concept. It showed that the brain's signals could be read and acted upon in real time, and it launched an entire field. Since then, the technology has advanced in ways that would have seemed like science fiction even 15 years ago. In 2021, a BrainGate participant who was completely paralyzed, a man who couldn't speak, couldn't move, couldn't even breathe on his own, was able to type on a screen at roughly 15 words per minute, just by imagining himself writing letters with his hand. The decoder was reading the neural patterns associated with each imagined letter and translating them into text. 15 words per minute might not sound fast, but consider that this person had no other way to communicate at all. And the accuracy was remarkable, around 94%, comparable to smartphone autocorrect. Meanwhile, Elon Musk's Neuralink has been developing a different approach, a flexible polymer electrode array with over 1,000 channels, implanted by a surgical robot, with wireless data transmission, so there's no connector sticking out of the skull. Their first human participant, Noland Arbaugh, received an implant in early 2024, and within weeks was using it to control a computer, play chess, browse the internet, all through thought alone. The engineering challenges here are genuinely formidable. You need electrodes that are flexible enough not to damage brain tissue, but rigid enough to be inserted precisely. You need wireless data transmission that can handle enormous bandwidth, 1,000 channels of neural data streaming continuously, while consuming very little power. You need algorithms that can adapt as the brain changes, because the brain is always changing, always reorganizing, always updating its own code. And this is where our earlier conversation about plasticity becomes so relevant. Remember how we talked about the brain's ability to reorganize itself, to reassign cortical territory, to learn new mappings between input and output? That same plasticity is what makes brain-computer interfaces work as well as they do. When someone first starts using a BrainGate system, the decoder is doing most of the heavy lifting. It's trying to figure out what the neural signals mean. But over days and weeks, something remarkable happens. The brain starts adapting to the decoder. The neurons being recorded begin to change their firing patterns to produce cleaner, more decodable signals. The brain learns to speak the machine's language, and the machine learns to speak the brain's language, and they meet somewhere in the middle. It's a genuine partnership, a co-adaptation between biological and silicon intelligence. But reading the brain is only half the story. What about writing to it? What about putting information in? This is where cochlear implants come in, and they're worth pausing on because they represent the most successful brain-machine interface in history, by far. Over 1 million people worldwide have cochlear implants, and most of us don't even think of them as brain-machine interfaces, but that's exactly what they are. A cochlear implant bypasses the damaged hair cells in the inner ear and directly stimulates the auditory nerve with electrical signals. It's literally writing neural code, translating sound waves into patterns of electrical stimulation that the brain learns to interpret as hearing. And the key word there is learns. When a cochlear implant is first activated, the signal sounds nothing like normal hearing. Recipients often describe it as robotic, tinny, like everyone is talking through a fan. But over weeks and months, the brain's auditory cortex reorganizes itself around the new input. It learns to extract meaning from this completely artificial signal, and eventually, many recipients can understand speech, talk on the phone, even enjoy music. The brain's predictive machinery takes this degraded artificial input and constructs a rich auditory experience from it. Helmholtz would have been fascinated. Retinal implants work on a similar principle, though they're much earlier in development. Devices like the Argus II, developed by Second Sight, stimulated the retina with a grid of 60 electrodes, giving blind recipients a very crude form of vision, patterns of light and dark, enough to detect doorways, follow a sidewalk, sometimes read large letters. 60 electrodes compared to the roughly 130 million photoreceptors in a healthy retina. It's like trying to display a photograph on a grid of 60 pixels, and yet people could use it because the brain is extraordinarily good at extracting meaning from impoverished signals. That's what predictive processing is all about. The brain doesn't need perfect data. It needs enough data to constrain its predictions, and then it fills in the rest. Now, there's a much more ambitious version of this writing to the brain idea, and it takes us into deeper territory. Deep brain stimulation, or DBS, involves implanting electrodes not on the surface of the brain, but deep inside it, in structures like the subthalamic nucleus or the globus pallidus. It's been used since the 1990s to treat Parkinson's disease, and the results can be dramatic. A patient with severe tremor has the stimulator turned on, and within seconds, the tremor stops. It's one of the most striking demonstrations in all of medicine. The electrodes deliver continuous high-frequency electrical stimulation that effectively modulates the activity of these deep brain circuits. Though honestly, even after decades of use, we don't fully understand why it works as well as it does. The mechanisms are still debated. It might be disrupting pathological oscillations. It might be activating fibers of passage. It might be doing several things at once. But it works, and it's been expanded to treat essential tremor, dystonia, and in more experimental settings, severe depression, and obsessive-compulsive disorder. And this is where things get philosophically interesting, in a way that connects right back to everything we discussed about consciousness and the hard problem. Because when you stimulate deep brain structures associated with mood and motivation, you're not just treating symptoms, you're changing how someone feels. There are case reports of DBS patients who, when the stimulator is turned on, feel a sudden lifting of the crushing weight of depression. Not gradually, not over weeks like an antidepressant, but in seconds. And when it's turned off, the darkness returns. What does it mean when your emotional state, your subjective experience, your sense of who you are, can be toggled by a switch? Some patients have described feeling like a different person with the stimulator on versus off, and struggling with the question of which version is the real them. This isn't abstract philosophy anymore. This is a person sitting in a clinic asking, "Am I still me?" And it touches on something Lisa Feldman Barrett's work would predict, that the self isn't a fixed thing discovered by the brain, but a model constructed by it. And that model can be altered by changing the inputs. Change the interoceptive signals, change the predictions, and you change the experience of being you. The frontier is pushing even further. Researchers at places like the University of Washington and Duke University have demonstrated rudimentary brain-to-brain interfaces, systems where one person's neural signals are transmitted to another person's brain through transcranial magnetic stimulation. In one experiment, two people played a collaborative video game where one person could see the screen but couldn't control it, and the other person could control it but couldn't see it. The first person's brain signals were decoded and transmitted to the second person's motor cortex, causing their hand to press a button. It's clunky, it's limited, it's more proof of concept than practical technology. But the principle it demonstrates is extraordinary, that neural information can be extracted from one brain and used to influence another. And then there's the question that hovers over all of this, the one that connects every thread we've followed tonight from ancient Egypt to Broca's laboratory, to Hodgkin's squid axon, to Chalmers standing at that podium in Tucson. The question is, as we get better at reading and writing neural signals, as the interfaces become more seamless and the bandwidth increases and the algorithms grow more sophisticated, do we eventually understand consciousness itself? Or do we just get better at manipulating it without ever truly comprehending what it is? Here's what I think is the honest answer, and it's the answer that most working neuroscientists would probably give you if you caught them late at night, after a couple of drinks, when they're not trying to sound confident for a grant application. The answer is, we don't know. We have made extraordinary progress. We've gone from thinking the brain was a cooling organ to mapping its circuits with nanometer precision. We've gone from Galvani's twitching frog legs to decoding imagined handwriting from neural populations. We've built computational models that can predict what you'll see before you see it, that can generate language, that can beat world champions at games of strategy. But the central mystery, why any of this is accompanied by experience, why there is something it is like to be you, lying here, listening to these words, feeling the weight of your body against the bed, that mystery remains. And maybe that's okay. Maybe that's even beautiful in its way. Because the brain that is trying to understand itself is using the very processes it's trying to explain. Its predictions all the way down. Your brain is constructing a model of your brain, constructing a model. And somewhere in that infinite regression is the feeling of being alive, the warmth of curiosity, the gentle pull of sleep. You've been listening to a story that spans thousands of years, from Imhotep's scrolls to Friston's equations, from a man named Tan who could only say one word to a paralyzed man who can type with his thoughts. And the story isn't over. It won't be over in our lifetimes. Every year, new tools reveal new layers of complexity, and every answer generates 10 new questions. And that's not a failure of science. That's the nature of studying the most complex object we've ever encountered in the universe. So let that settle. Let the questions drift. You don't need to hold on to any of this. The names, the dates, the mechanisms, they'll be there if you want them later. What matters right now is that you followed the thread for a while. You let your curiosity wander through this extraordinary landscape, and now your brain, that 3-pound prediction machine that has been modeling the world and constructing your reality all day long, is getting ready to do something it does remarkably well. It's getting ready to let go, to quiet the predictions, to soften the error signals, to stop updating and simply rest. The thalamic gates are closing. The cortical chatter is fading. The slow waves are beginning to build. And you, whoever you are, wherever you are safe and comfortable and exactly where you need to be. The brain will take it from here.